Hey Malhar, welcome to the second episode of the Token Economy podcast.
Yeah, thanks for having me, Shaurya.
Totally. Let's get right into it. So, why don't we start off with you introducing yourself and origin and what you're building?
Yeah, I'm Malhar, as you mentioned. I grew up in Bombay. I studied computer science at the University of Illinois, Urbana-Champaign, which is in a very small town, 3 hours away from Chicago, but famous school, I think. And I'm basically the co-founder and CTO of this company called Origin. And we're doing AI for biology with the application of being able to make these synthetic DNA switches that sort of program gene expression very precisely with the sort of long-term goal of making gene and cell therapies much safer.
Awesome. And I've been so excited to have you on the podcast because I feel like we've had so many of these conversations offline about how you're using AI to decode biology. So I'm so excited to have you on. But let's get started from really the beginning. I know you grew up in India just like me. So talk to me about what was childhood like in India and what drew you to biology?
Yeah, childhood was great in India. I miss it a lot. Not being in India, I'm going back in a few days, so should be nice. I think just growing up, I mean, had very sort of an academic childhood, which my parents were not academics as such, but they did enforce a lot of academic tendencies, and I took like my education very seriously, or at least they made me take my education very seriously. My sister was like always a much better achiever than me in school. Like she would always have like really, really good grades, and I had terrible grades. So it was always like following in her footsteps, which I never managed to do, but I was sort of told to do. I think within that entire sort of experience, I just took studying seriously. I just read a lot of books. Growing up, it used to be just like fictional books, but then eventually non-fictional books. And I was a lot more interested in programming than I was in biology growing up. I used to play like Minecraft and I had like a Minecraft server, and I had to convince my dad to like pay $10 a month to host a Minecraft server, and I couldn't convince him to pay for like a Java developer. So I had to learn basically Java to make these like plugins for Minecraft servers, and that's how I got into programming. I was always interested in biology, like my dad's a doctor and my sister's in med school now, but I was always more interested in programming. And eventually, the reason I got into doing what I'm doing is because of all of these weird analogies between biology and computer science. That's why it's compelling. So it wasn't as much as the biology growing up, but more so just the computer science of things.
That's pretty cool. And you told me this funny story just last week at our dinner that you and Yash dropped out of school. I think you had a year left and Yash had a semester left. Most people at that point just end up finishing school, but clearly you decided to drop out and start Origin. What was the catalyst and what made you feel ready?
Yeah. I was always open to the idea of dropping out. Like it was actually one of the easiest decisions I've made. Like it was literally over like one coffee that we were like just might as well do it. And even with my parents, it was like very easy to convince them. I think I was thinking of it for a while, like even before starting Origin, it was just to drop out and go work for our common mutual friend Govind. And I think a big reason for it was I spent like my summer in the Bay Area. I just saw how fast things were moving here. I think I just saw the models also getting much, much, much better. And like I was never of the opinion that like school is bad or to like it was not never a decision that I want to get away from school. It was more so that there was just so much more to do and like much higher pace and speed and things just in a much better way outside of school. And I think Origin itself, like it wasn't just any company, like we really wanted to work on these biomodels. So it wasn't like we'll drop out and we'll figure out what we want to do. It was that we wanted to do this, and there wasn't any sort of inhibition on the fact that we had to drop out. And I think like I dropped out with like a year left. My co-founder Yash dropped out with like a semester left. I think it was still too late to drop out. Like we should have dropped out much, much sooner. And I think my opinion on it has changed now. Like I think when we dropped out, I would have had like the opinion that maybe it's not for everyone, and there's some merit in still staying in school only if you want to do your undergrad degree. I think like I have much more radically charged belief now, which I think is like completely useless to stay unless you want to like become a doctor or you want to go to like some very regulated field or you want to do like a PhD. I feel like if you don't want to do any of that, like I just cannot imagine why you'd want to stay. And I think it's at a point where it like actively hurts you now. Like if you wanted some hot takes for your podcast. I think that's one.
That's definitely a hot take. Talk to me a little bit about you're of course talking about kind of dropping out of school and how you feel like everybody should. What do you think stops people from doing that? Because you're totally right. It feels like in Silicon Valley, especially in the last 12 to 18 months, the pace of innovation is just so rapid, and you almost feel the palpable energy. It's like you get out on the road and you see all these people running around, and you go to coffee shops, there are meetings everywhere. The energy is so palpable. Why do you think people don't do it?
I think there's one aspect of it is just like lack of surface area exposure to people, to a group of people who actually do it very actively. Like if you're in like I imagine if you went to like Stanford or like Berkeley, that's like you're gonna have a much higher rate of people dropping out. I'd assume. I felt like at least in UIUC, it was so remote. And like Chicago is a great city. I think it's one of the best cities in America, but like there's like zero tech scene there, and people aren't really dropping out for like if they want to go into finance, for example. So I think there was one aspect of like no surface area or that sort of helps you build the conviction and it sort of like makes it normal. And I think the other aspect of it is also just very poor sensitivity of like a response to changing changes in technology. Like I think it has not clocked people like how fast things are happening. Like imagine you were joining computer science degree right now and like your goal is to become like a front end developer. Like I don't know if there are people like that exist today, but like it's sort of like what do you think is going to sort of be the outcome of that? And I also think people think that when you drop out, it's sort of like being in like you have to have figured out what you want to do. Like I think with dropping out also and working on the company, like if you have conviction to see the company through, it's also a good time just to do exploration. Like if it's like if you're doing a research company, for example, like a lot of the things you're going to learn when you're in a company setting. It might be because you don't have compute before, like you're working with like a cluster of like 10 GPUs from your university shared by like 1,000 people. So I think there's just so much unlock, and I think people just don't realize that.
Yeah, no, totally. That makes a lot of sense. And on the part of exploring, how do you even come up with the idea of origin and the idea of using AI to build these
Yeah.
models?
Um, I think with AI, it was sort of like the when I mentioned earlier, there was this sort of analogy between biology and computer science, and specifically AI, that we found quite interesting, both Yash and me. I think was that if you look at like evolution, evolution is sort of like an upper bound on like the slowest and like the worst possible solver for a program that does what we do today, like what intelligence is today and like how that has sort of grown. I almost felt like AI was the exact opposite, where you're using these like gradients which are telling you the directions to move in. It almost felt like if you're doing like sort of bounds, like one was like fastest possible one, and one was like the slowest one. It's not entirely true. Like I'm sure there might be something better than using gradients and doing gradient descent the way we do it, but it almost felt like very natural to want to apply AI to do things in biology because we know that biology has like grown in the sense of how evolution has made it like grow and evolve and applying AI to do that on like a philosophical level feels very natural to how we got here in the first place. I think the other thing was just watching, like on a more practical lens, just watching models get better. Like obviously the first introduction to AI for in biology for like me and probably pretty much everyone I know was like AlphaFold. Just like seeing the results of that, and if you see how people used to do like structure prediction before. Like I was just reading a book on a Nobel Prize laureate who sort of won the Nobel Prize for discovering the structure of the ribosome, which is like this protein complex that actually converts DNA into sorry, it actually just takes like nucleic acids and RNA and it makes it into proteins. But it was sort of like it was such a tedious process and such a long process. And it's sort of insane that with AlphaFold you could just do a lot of this and you can just speed it up. And it's AlphaFold's not perfect, but it was still such a step change in ease. And it felt like we've seen that. We saw that if you wanted to apply it to more things like sequences, like amino acids, like nucleic acids, we saw that on sequence sides, language models are getting better. So there's almost like proof of existence on so many different components. And it felt like we could just make the models, like we could train better models for biology and in this DNA regime than people were doing. I think working at like Automorphic, I was just like exposed to a lot of cooler ways of training these models, improving these models, and just felt like when is someone going to do like some of the stuff you were doing at Automorphic for like biomodels, it just felt like light years away. So that was like one of the technical inspirations for it.
No, that's really fascinating, and it feels like biology is, of course, like this really complicated, but also this really broad field, and you've kind of narrowed down the problem of what you want to do. So, I guess tell the audience a little bit about what was the first wedge in terms of like when you start the company. What was the first thing that you all end up thinking about building, and how did you even come about? Like, what are the initial results that you need to see in order to pursue, in order to basically go out there and pursue this thesis of like controlling gene expression?
Yeah. I mean, we definitely didn't start with exactly this. And I think even from now, it's going to sort of evolve to increase the scope of what it is. When we started, we were just interested in the idea of being able to do better, like CRISPR, which is like this gene editing technique where you like knock out a gene. And it felt like one idea we had around there was playing around with engineering better enzymes, these enzymes called Cas enzymes, which actually perform the knockout, like playing around with things like the size of the Cas enzyme, playing around with even like this idea of how do you get reduced off-target effects, because when you actually perform a CRISPR experiment, it can sometimes knock out genes that you didn't initially target, and that has all of these safety issues. I think there's some aspects there about what is the need for who has actually already worked on improving the Cas enzyme. But that was our original idea, which was it was always about the genome. I think we just found the genome quite compelling because it's almost like this codified. It's like the only proof we have that intelligence in some sense can be like codified, like it can be written down. Like that's the only thing. Like we know that that exists. Like obviously to decipher it and like the complexity of it is one thing, but we know that that's there, and that's what we were always just like philosophically drawn in by. And we started with like gene edits and CRISPR. Just spoke to more people, just spoke to a lot of people, figured out like what people want, what people need, and we sort of struck or like stuck to the DNA side. But we tried a whole bunch of different things. We tried training like these bacterial genome models, tried training different like single cell models, like gene expression models. I think the main thing for us in our exploration, especially for like the first year, was just getting like our like deep learning training muscle much stronger. Like we like pre-trained like 10 billion parameter models with like barely any compute. And I think that was like the first year of just like focus of just like training, setting up the infra to train these models, like setting up the infra to do good, quick experimentation. And in that process, just speaking to a lot of people, seeing like what really mattered. And I think there's things about biology that we learned that we didn't know. Like for example, translational risk of what might work in mice, what might work in like cells, but will not work in like humans. Like what really moves the needle. I think like that's those are things we learned when we spoke to like pharma companies or biotech companies and told them like, give us money to do this, and then they were like, it doesn't really matter to us at all. But yeah, I think that's it.
Okay, that makes a lot of sense. And you just talked about kind of data and like kind of all of these this massive data set that you're producing. I guess can you help expand on that in terms of like one, how are you generating this data set, and then what is the scale of this? Because you just mentioned you trained this 10 billion parameter model, which of course is insane for the amount of compute you have, but along with that is of course much smaller than all of the I guess the cutting edge models that we're seeing. In terms of how do you think about the data you're producing, how do you train the model, and like what makes this very defensible and unique?
Yeah. I think the biggest defensibility from like makeup side is definitely the data. Clarification, this model is actually insanely small. It's not even 10 billion parameters. Like it was just like experimenting, pre-training 10 billion parameter models and north of 10 billion parameter models. This model is like sub 100 million parameters, like the one we have right now. So it's really, really cheap. We sort of designed to make it that way because there's an aspect of the model generating a lot of candidates, like candidate DNA sequences. So we do a lot of work on the test time as well. So let's say we want like 100,000 sequences that we want to test out, or we want to provide just like a customer. We like use the model to generate like 100 million sequences, and then we actually filter a lot at like test time, at sample time, and we'll spend a lot of time doing that. And how fast we can do that and the scope at which we can do that just depends on, or it really is helped by the fact that the model is very small. When we also look at these DNA sequences, regulatory DNA is very short. It's like 200, 300 tokens long. So you can have very, very high batch sizes. So if you have high batch sizes because of short context lengths and you have like a very small model, that's why we can actually just generate so many sequences. And in terms of the data we're making, I think we're interested in like two types of data. One is like far more direct, which is what we're immediately starting with, which is we take these synthetic DNA sequences that we make, and like some context on what these do is like your genome, you can think of it like there's certain parts of the genome that actually code for a protein. They actually make the protein that goes on to perform most of the functions that your cell does. And this part of the genome that actually controls where these proteins are made and when they're made and just the regulatory aspect of it essentially, which is sort of why like one of the reasons why like different cells in your body have the same genome, they have like coding regions for the same proteins, but different proteins are made. Like your neurons do different things from like your skin cells. It's because the protein that codes for like or the DNA that codes for neuronal proteins, for example, is actually activated by these regulatory regions. So one of the direct experimental things we're doing is we're actually designing these synthetic regulatory regions, which are like these 200, 300 long ATGC strings that are not found in nature, and within the generated set are quite diverse. And we're targeting them for specific cells. So we might say, okay, I'm generating 100 sequences that I want to target for cell type A. And what target means is it should only make a gene in cell type A. So we'll take those 100 sequences, we'll put them in three different cell types, let's say cell type A, cell type B, and cell type C. And we want to essentially show that how much of the gene is turned on by this regulatory sequence intended for cell type A, that amount should be much, much higher than how much the gene is turned on in cell type B and C, which are termed as these like off-target cells, for example. So what we're doing is instead of 100 sequences, we're taking like a million sequences. We're doing like millions of sequences, actually targeting different cell types, different combinations of cell types. So it might not just be one on, everything else off, two on, everything else off, for example. So you almost get like a power set out of all of these things, and just checking and seeing where did these sequences turn on these genes, getting that actual wet lab data. So actually conducting these assays, these like MPRA, which like the massively parallel reporter assays to study this. And we're just getting the data on that. So we get the actual synthetic sequences that our model generated, but we also get real biological verification of this. So we're not using like a other model to do predictions. So far, we've been using other models to do like forecasts on what to expect, and that's how we select our initial library. Um, but the idea is just to test millions of these sequences. We're starting with these like immortalized cell lines, which are cells that have been made to be able to replicate faster. And they're not the best in that they have a very low rate of actual transfer to clinical outcome, like even not even clinical outcome, even way before that. So they're not very useful for planning things you want to do later on. But they're just a good start. They're cheap, they're easy to make, and that's what we're starting with. But then we're also looking at actual primary cells, which are cells derived from humans, and those are more therapeutically and clinically relevant, essentially. So we're starting with this and just testing millions of these sequences. That's one aspect of the direct data collection process. But another aspect of it is we're working on getting a lot of uh, actual tumors derived from patients. So we're speaking to some hospitals, and we're trying to actually get a good supply of tumor resections that have been received from actually performing major surgeries to sort of get rid of cancers. And we're taking those tumors and we're sort of performing experiments where we inject our synthetic sequences directly in cells in those tumors. And that idea is to get a lot more sort of clinically relevant patient data and actually seeing how people would respond to genes being, or not people because it's still outside, but how more relevant cells and immortalized cell lines would respond. And the aspect we see is with a direct set where we test out sequences that work well, we can actually just license these sequences or we can use these sequences. Like imagine we got a sequence that turned on a gene very selectively in my target cell type and not in other cell types, just licensing those. And it's almost like the end goal in its own. But the other thing we see with a lot of patient tumor data is just using the sequences as a medium to study these cells, which
Mhm.
is let's produce a very diverse data set where we perturb certain cells to express certain genes. We see how that cell changes and we get very, very rich information. Now, this information might be beneficial to train much better models that are better at predicting, that are better at discovering new targets, that are better at predicting what a patient's response is going to be, what my dosage should be. So a lot of like clinical stage stuff as well. And that might be a bit more indirect, where it might not be that I want to do anything with that sequence, or that sequence is the asset I want to sell or to license or to use, but more so that it just allowed me to induce a particular state in the cell by selectively expressing a gene, and just using that and using like hundreds of thousands of sequences and using hundreds of thousands of different states and just getting a lot of data from that. So I think those are the two angles of just data collection that we think will be the biggest modes.
Okay, and who ends up being like clearly there are so many ways in which you can monetize this data, the model, and then there are a bunch of different pieces. Who actually ends
Yeah,
up being kind of the core customer, and where do you see yourself in kind of the life cycle of pharmaceutical companies, and also just in terms of like as a company that builds this model but also has the data, who ends up being the end customer?
yeah. I think on with the sequences, uh, like a lot of people we've spoken to and speaking to are these biotech companies that are working on, let's say, gene therapies. They're expressing some sort of gene, and they wanted to selectively express it in particular cells. So they would be interested in, let's say, hypothetically, just taking the sequence itself. So we test this out. We give them the sequence. They pay us some upfront capital. They might pay us when they start clinical trials. They might pay us to actually license the sequence to use in these trials and use in the drug. So our sort of goal at that point ends at just providing the sequence, having tested out the sequence. On the side of the perturbation data, it's a bit more open-ended, where it could be bigger pharmaceutical companies. So we could train models on that data and license the models themselves to these pharmaceutical companies. Like, for example, there's a company called Noetic that does this really well. They basically train these single cell models and a lot of multimodal models on tumor resections. And they just got like a deal with a big pharma company called GSK that I think is paying them like I think like $50 million a year. Um, I think some ballpark that range, but just accessing models that have been trained on like a lot of this proprietary data, especially in like from human derived tumors. And so that's there's an aspect of that, which is just training these models on proprietary data, on relevant data, on clinically relevant data, and allowing people to use those models and licensing those models. So I think there are those two aspects. And I think the data itself can also be valuable to people. It can be valuable to pharma companies. It can be valuable to AI labs because it's very rich data on like functional data on how cells function, like the behavior of cells, like responses in cells to some sort of stimuli. So I think there's an aspect of the data in itself also being somewhat of a valuable asset.
Yeah, and how much of the data that you produce, because you kind of have these test cases, but then there's also like these specific cells that you're of course targeting. What percentage and what percentage of these end up in the wet lab for actual experiments, and how do you even determine what gets to the wet lab? Because clearly you have billions and tens of billions of data points.
Yeah, on the side of when we're testing these sequences in the cells, I think there's a lot of things we consider, which is like on terms of the sequences, we run like a very systematic process on evaluating sequences we generate. So we use off-the-shelf models that people have already trained and tested in labs to actually predict the experimental outcome. So it's not going to be perfect, but it's sort of like we use a model that, let's say, they tested in a lab in a similar setup to what we want to do, and they had like zero point 0.9 correlation with their model's prediction and actually experimental readouts of when they perform these experiments. So we use that to actually predict how our sequences are going to function, and then we have these thresholds and we choose things. So we might have a sequence where it has, let's say, 2 log-fold change higher expression in the target cell versus the off-target cells, like the max of the off-target cells, and we'll use this predictive model to basically choose that. And so we do a lot of work with predictive models that are independent of even our models to sort of figure out what sequences we want to take, and just a lot of predictive work in ensuring that most of the sequences we use do what we want them to do. I mean, obviously, we'll see the outcome of that when we finish running these experiments, but that's the sort of goal. And there's a lot of things on the manufacturing side also. So the sequence might be great, it might work theoretically, but the sequence actually needs to be synthesized, and they use enzymes to synthesize this. And sometimes, for example, if you have too many G's and C's, like if you have ATGC in your sequence, it's not favorable for synthesis. There are all of these other things. If you have very long sort of strings of the same nucleotide letters, like imagine like 10 characters of like just A, again, it's not very favorable for synthesis. So we do a lot of quality checks on that end. That's how we filter things. We also do filtering on novelty, where I might generate 100 sequences and they might be very similar. So I just have very, very little like unique information content in that. So there's a lot of diversity checks that we do as well. So I think that's how we sort of select sequences. And in terms of cells, it's just figuring out, ensuring there's enough cells that we get like statistically significant results. Because when you use, like we basically use viruses to actually get these sequences into cells, and sometimes the viruses might not get into cells. So there's a lot of like layers of failure that can happen. I think that's one of the biggest lessons we had coming from a computer science background is that like it's like theoretically everything sounds like incredible,
Yeah.
and like you like love theoretical guarantees from like a computer science background. But I think just even though we've just started running these experiments, or we've just sent our first library for DNA like synthesis, um, just working with data, uh, available data, we've just seen the biology is like very noisy. A lot of things fail. So a lot of it has been just making sure the experiment is robust in its design.
Yeah, no, the real world is messy and noisy, and I feel
Yeah.
like biology is probably is probably the best example of that. And one of the things is the fact that we've kind of seen this real acceleration in applications of AI in science. And of course, you're kind of at the frontier of this and at the cutting edge and are working on this every day. I guess as you think about where models are today, and of course, there is like one is just LLMs being applied to biology, but then there's also multimodal models and other models. I guess like what are you most excited about in applying AI for science? Of course, other than what you're working at Origin, but what excites you more, and why do you think we're seeing a lot more acceleration now versus 12 or 18 months ago?
Yeah, I think what excites me a lot is these automated labs. I think OpenAI had a collaboration with Ginkgo recently, where they just basically like hooked up a model to be able to do like automated lab work. And I think that's really, really going to help things take off, because at some point you want these models to actually explore with the physical world, to conduct experiments. If you have a model and you've really optimized the model, and the model can produce thousands of tokens in very short periods of time, you're obviously going to get like rate limited by a human doing these experiments. So I think I'm quite excited about automated lab work, because like lab work is obviously sort of bedrock of so many things, and it could be in biology. It could also be the kind of work periodic is doing, where they're trying to go for like superconductors. At least that's what I think they're doing. And I think that also just requires automated lab work. So I'm quite excited about that, in just like a generic science perspective. And I think the reason that's going to probably be the most fruitful thing is if you look at like language models working really well because the internet existed. Like I think the biggest sort of bottleneck in science so far, and even I think in I guess in robotics, is just like data not being as abundantly collected and present as it was in the case of the internet. And I think again, the reason we're going to see acceleration in this is because probably just a lot more capital investment, knowing that it's almost not entirely de-risked, but the technical idea and the technical conviction behind it has been de-risked by the fact that it worked for language models. Like we know that when we had like this very rich repository of probably a lot of even like brain rot on the internet. Like I don't know if they like filter like a lot of Reddit stuff when training these when pre-training models. But in spite of that, like these models are they're so malleable, and they're so good at actually fitting these functions. And if it worked on the language side, like the model's just saying numbers, like of course there's patterns to these numbers. Like there's no reason why it will not work on the science side if you can actually sort of orchestrate all of these surrounding things of being able to get the data and supplying the data and reducing like these feedback loops. I think that's also going to be an important thing. Like if you look at like code increasingly becoming obsolete, like especially like even in like the last 3 weeks, like that I feel like there's been a step change in code with the new models. Um, a lot of it probably is because code is just easy to verify. Um, Even people like accepting or rejecting suggestions on like Cursor, that probably just give like good data on like what's right and what's wrong. I think even as we get automated labs, the main aspect is going to be how do you ensure that there's very quick feedback. And I think if there's very quick feedback and we have these automated labs, like I think it's going to get like I think it's going to take time. There's probably even hardware constraints to it. I don't think it's going to happen as fast as code happened, but I think it's definitely going to happen.
No, totally. And from my perspective, the opportunities, of course, in the physical world are far greater than the digital world, even because we've started to automate a good chunk of the digital world. It's still very early days, but in the physical world, it's even earlier. And you're totally right, which is, I mean, basically what we're talking about is just scaling laws, which is like, can we apply scaling laws to sciences, to the physical world, and really to biology, and see, can we solve cancer, and can we solve all these diseases that have been unsolved for a very long period of time, and just throw lots and lots of data at it. And here's an interesting question, which is, on the compute side, as a startup, that of course, like you mentioned, you're training these very small, very cheap models, but along with that, as I mean, the whole perspective behind scaling laws is that you basically throw more data, more compute at these models, and you just scale them up really quickly. How do you think about compute, especially in an environment where, of course, we're seeing the largest players like the OpenAIs, Anthropics of the world, and all the other top 5, 10 labs just sign massive multi-billion-dollar, tens of billions of dollars worth of compute? How do you see compute as a bottleneck for Origin?
Um, I don't think right now. I think it's for what you said, which is like specialized use case. Like I think if you look at these language models, I'm like very pro scaling. Like I think I've always been very pro scaling. Like I think I just find it insane when a lot of people think that scaling is not the right direction because it doesn't seem like nice. Like you want to make algorithmic breakthroughs, and you want to like have something more complex. I've always found like if like the way you're trying to make an intelligent system, like if it doesn't scale, like it's obvious that like your building blocks, like your axioms are incorrect. Because if it doesn't scale, whatever's like your smallest computable unit, like it doesn't have like a property of like actual like compositionality. Like you can't combine these things. There's no like inductive process happening. So that way I'm like very, very pro scaling. But I think there's very complex relationships in scaling between when is something underparameterized, when is something overparameterized, how much information content is there in the data, which directly informs like how many parameters you would need to sort of compress something. I think on scaling, I think it's very important from like a AGI perspective. And I think in that sense, the models have done really well. Like even when people say like models don't learn as efficiently, like I think on a temporal scale, they're probably right. And from like a how heavy it is, like you can teach someone, like I can tell you like 10 things and you can probably learn them. And with like GPT, that probably won't happen. But I've always found it to be a bit insane that if you look at GPT-4, like even just GPT-4, I think came out like 2020 three, I think came out 2023, like a trillion parameters. And the breadth of knowledge it has is so insane. Like it was like decent, maybe like 65th percentile in like law and like medicine and like a whole bunch of these different things, like math and coding. And it had probably, I think, like two orders of magnitude less parameters than I think the human brain does. If you take like human brain as like let's say 100 trillion parameters on estimate, like I don't know how correct this is, but I think it's so insane that with 100 trillion parameters, you don't like you're not 65th percentile in like almost every field that exists. So the actual amount of like bits that these but like information bits these parameters are storing just seems to be so high. And I think that's going to have a lot of benefits in scaling AGI, which is why like I think like the capex investment in data centers is like not enough. Like I think it should be far, far more. Like we could have lived in a world where things didn't scale and like what do you do at that point? And like it's so obviously scaled till now. Like I can't imagine why it would stop. Like there's something so fundamental about the structure of it that is like building on top of what was there. And it should continue. And for our sense, I don't think it's a problem immediately, but it is something we like think about in terms of getting access to more compute gradually. Because if like we believe so heavily in scaling, there's some effect of it when we start getting a lot more data. Like as we start collecting data from these patients, as we scale that up, there's an aspect of scaling that up first from like a logistical perspective and a physical perspective, which is probably more important in the short term to figure out than scaling the compute. But I also think like scaling compute is probably going to get cheaper, like compute's going to get more available. Like there's going to be all these downstream benefits of like more data centers, more energy. At least I hope there is. And so I think it's a long-term thing you would want scaling because you can keep pushing the frontier with it. I think for a lot of our objectives and even objectives in actually bringing like drugs to market and actually helping pharma companies and biotech companies do that, getting models that are good in the clinical stage that can predict patient response, that can help with patient stratification, that can help with like actual functional things such as like making having good dosage, like figuring out how much of a drug you actually want to sort of use. I think we don't need like crazy scales for that. And I think what we have, and even the compute we have available, is sort of adequate to have something that's capable of doing that. So I think in terms of practical use immediately, I don't think it's as much of a bottleneck.
Yeah. And one of the very interesting questions that I've been pondering over is AI has, like you mentioned, the amount of intelligence that we have really in our pockets now is pretty unbelievable. And the next step, really, in terms of advancing science, is when does AI start to actually make novel discoveries and come up with new things? And it feels like we're really on the verge of it. What are your views on when AI starts to develop novel things for biology and across the sciences?
Yeah, I think across the sciences, if you like count math, and sometimes it's already started doing that. Like it started solving so many of these open problems. Like I think I saw a bunch of like these new math startups. I think like Axiom is one of them, and they like solved a lot of these problems that have not been solved. I think it's been really good at like literature review. Like I remember seeing, I think a couple of months ago, like GPT being really good at being able to synthesize information that's dissipated in like a lot of different papers. And I remember like I think people like shat on that because like I think OpenAI said it made like a new discovery. But I think like that is also just really important work. Like when you have like hundreds of thousands of papers and a lot of things you can probably figure out just by figuring out how these papers are relevant to each other. So I think in that sense, it's already started. I think a lot of like the blockbuster scientific discoveries will first happen in math because there isn't this physical constraint of having to run physical experiments. Like you're not rate limited by the sort of slowness of that essentially. So I think maths will be first. And I think in biology, I think it's already starting to happen. Like I think Goodfire, like the Meckendorf company, they recently did work where they showed they were able to sort of figure out some sort of understanding of I think it was in Alzheimer's. I think these are going to happen slowly and slowly and slowly, almost like coding was happening earlier, where you could like get like a function to be written, etc. But I would think like maybe like in a year and a half is like the infrastructure for these automated labs gets better. Like I think we're going to start seeing it happen very, very rapidly. I think in terms of even the work we're doing, I think we're going to get to a point where the amount of synthetic sequences we have that are able to actually induce certain cell states is going to be so much larger than like what is like mined from the genome that exists and from like different people's genomes that exist today. I think that's slowly going to start happening, but it's going to happen really quickly when it actually starts happening in somewhat of a consistent way.
Yeah, no, very cool. And let me ask you the last question I ask every guest, which is, what's your wild prediction for 2026?
I think my wild prediction for 2026 on this note is I think we're going to solve a millennium problem using AI this year. And I would actually bet on it being entirely autonomous.
Wow.
Think were like I've seen some reports of like DeepMind being close to it anyways. And I think if you see the recent results in these models and how well they've performed at the IMO and the IOI, I think even some of the OpenAI stuff where like they had like the same RL system. So clearly some improvement in not having these very like bottlenecked funneled rewards. And I think we're going to see a Millennium problem solved this year. Actually, maybe not autonomously, because I don't even mean not autonomously from the fact that it wouldn't have the capability to have done that autonomously, but probably people started working on it in conjunction with the AI, so you can't really claim it was autonomous and you couldn't prove that it was autonomous. But I think this year is going to be a Millennium problem being solved.
That would be pretty unbelievable. And I have to say, this conversation has been pretty epic, and I feel like I'm walking away so much more energized and optimistic. So thank you so much for coming on.
Yeah, thank you for having me. This is my first podcast, and I hope it's my last podcast because I don't like going on podcasts. But
So glad to have you on.
yeah,
Awesome.
it was great.